In this presentation, we're going to look at actually how the muscles contract. We will begin looking at how the muscle fibers get a message to contract. The muscle needs a lot of energy, and we already said that if all cells get energy saved in ATP by cellular aerobic respiration. So this is also the primary source of the muscle, but it has two other sources. It produces creatine phosphate, which can be broken down into creatine phosphate and some energy that can make more ATP. And then thirdly, muscle cells store glucose in the form of glycogen. So here again, this glycogen then can be converted back to glucose for cellular aerobic respiration. But it is a third option that has an extra source of glucose. Since we remember our formula for cellular aerobic respiration being simply glucose plus oxygen yields energy and ATP plus the byproduct carbon dioxide, which the body needs to get rid of, plus heat plus water. And those last two things are called byproducts because the body can't use them. So we need oxygen. In order for muscles to contract, they need a lot of oxygen. And there are two sources. Just like every other cell, we breathe it in from the outside, it comes into the lungs, and then it gets transferred to the blood, and it attaches to the red blood cells, the hemoglobin in the red blood cells. So that's one source that every cell has. But muscles, since they often need lots of energy, also store oxygen in the myoglobin. Just some terms that you might have heard. Oxygen debt, that's when there is insufficient oxygen when we're having strenuous exercises. Glucose is converted to lactic acid, and that gives that feeling of muscle fatigue, that achy muscle. And then we talk about recovery oxygen uptake. The lactic acid then gets converted into pyruvic acid using ATP. And pyruvic acid is a part of energy production as well. So when you stop exercising, your breathing rate doesn't decrease right away, and your heart rate doesn't increase, because here again the muscles still need to keep working until this process finishes. So now we will look at exactly how the muscle works. This will just be the beginning, but we can see the muscle fiber in the hand, or the muscle in a hand, is lots of fibers. That's the next towards the right picture. And each muscle fiber has these little fibrils in it. And that's what gives the muscle, the voluntary muscle, the striated appearance. Then we can see that these muscle fibers of the muscle cell are encased in a coating called a sarcolemma. And right underneath it is something called sarcoplasmic reticulum that is full of calcium. We'll talk about that in a bit. We'll see the two T-tubules go down inside. We'll talk about what that does. And then a little bit in the next presentation, we'll see how these filaments actually, within the fibril, one pulls one over another to contract. But we'll look at more at that in the next presentation. So we said each muscle cell, we've said this before, muscles need to get a message from the nervous system. So each muscle cell has its own motor nerve. The axon terminal, meaning the end of this nerve, and unfortunately we will study nerves in detail a little bit more in the next chapter, but nonetheless, they have a neurotransmitter, because once this message is going to leave that nerve, it's a chemical message across a little space called a synapse that has to give the message to the muscle. And the neurotransmitter in the neuromuscular unit, which is the nerve for the muscle and the muscle itself, is acetylcholine. And we said earlier when we looked at the picture that the sarcolemma was the membrane that encased the muscle fibers. And this has receptor sites for acetylcholine. So then it can accept this message. Now it can only take a message periodically. If it had a constant message, we said later, the muscle would just fatigue itself out. So the inactivator is cholinesterase. And as we'll talk in the nervous system, a lot of the message through the nerve is electric. Well, there's a bit of electrical part also that gets the muscle knowing to contract. And in order for that to happen, the muscles need to have this state of electricity. So we say it is polarized. It has a resting potential. So it is polarized when it is relaxed, meaning it can do something with that message. And then, as I mentioned earlier, the space between the axon terminal of the nerve and the sarcolemma is a little space called the synapse. And then the myofibrils are like long cylinders, but they are broken into little contractile units called sarcomeres. There's a whole line of them. They are bound on either side by a Z-line, and inside of them are myosin, the contractile protein, and it's in the center. And then at the edges is actin, also a contractile protein, and it is attached to the Z-lines. And this actin also contains inhibitory proteins called troponin and tropomyosin. So we can call them inhibitory or relaxing, because if they are active, the muscle cannot contract. And as we mentioned before, the sarcoplasmic reticulum is right under the sarcolemma, the coating. It surrounds the entire sarcomere, and it has calcium, because we're going to see that calcium plays a very important role in muscle contraction, and it has to be available, and this is where it's available. Although, remember, calcium was stored in the bones so that it would be available for other body processes. As we said before, a relaxed muscle fiber is polarized. It has a potential, and it does use energy to stay this way. The outside has a more positive charge. Sodium is on the outside. And if they get their way in, we call it the sodium pump. That's the process that uses energy to push them back outside. And then on the inside of the sarcolemma, there is more potassium. And here again, potassium needs to stay inside. So we have a potassium pump that will take care of that as well. So when a muscle is told to contract, we want the activity called depolarization of the sarcolemma to occur. So the nerve's impulse message arriving at the axon terminal releases acetylcholine. And acetylcholine, as we said, has to bind to the receptors on the sarcolemma. And when that happens, then the membrane becomes more permeable to sodium, and they rush inside the cell. Happens to make the cell more positive. But this change is called depolarization. This is the activity that gives the message to the muscle to contract. And it spreads along the entire sarcolemma, so it's kind of got to move along. And we mentioned those T tubules before. This carries that action potential inside. And when it gets this message of depolarization, as I said, it would contract. And we're not going to go into all of the chemistry of it, but something has to stop that message, because it cannot continue or the muscle fiber would just wear itself out. So there is a substance called cholinesterase that can appear at that synapse to inactivate the acetylcholine to stop the message. And here we can see the picture of the tan at the top is the axon. The neuron, the axon comes down in the darker tan, and then the lighter tan would be that axon terminal. And you can see there's a little vesicles of acetylcholine. So if it gets the messages to tell the muscle what to do, they're going to open up, and the acetylcholine is going to go across that synapse. And those sort of deep impressions are the T tubules, but the little dots, I don't know, on the pink, little ovals, I guess, those are the receptors. So they must accept the acetylcholine. And we said when that happens, then this sarcomere, the sarcolemma, the lining, depolarizes, that the sodium comes inside. It tells the muscle it can contract. And I think that's what it's showing you on the bottom, the sodium coming inside of the muscle fiber. And as we said, the muscle fiber cannot continue just to get messages. It needs to stop. So the stopping message is called repolarization. When the acetylcholine is inactivated, potassium runs quickly to the outside. And here they are, they're reversed. And as soon as they reverse the other way. That stops it, and it leads back to the polarized state so that the muscle is ready to contract again. The sodium ions, like I said, are normally on the outside. Potassium are in the inside. That means it's polarized, it's ready to go. And it can then get another message. And here we see a visual of that. So we see the sodium on the outside, the potassium on the inside. We can't see the A above this, but that says the polarized state, the ready to go. And then in the depolarized, we see the acetylcholine coming in, and then the sodium going inside. So when the sodium is inside too, that's repolarization. Excuse me, that's depolarization. That's the message. And then the bottom one, when the message stops, potassium go to the outside, and you can see they're exactly opposite. And that just happens very quickly, and then it goes back to the polarized state so that it's ready to do it again. So take some time and think about how the muscle fiber gets its message from the acetylcholine, from the axon terminal of the nerve cell, and how it changes these ions. It's in a polarized state so it can go, but getting the message is depolarization, sodium coming in. When the message is done, acetylcholine gotten rid of by cholinesterase, the potassium goes to the outside, and as they're reversed, they'll quickly reverse back to the way they were of sodium outside, potassium inside, polarized, so it can go again.